So I will tell you about, it's an overview essentially about our big data work with the concentration on Exposome. So just a recap, I think a lot of what you heard already, our health is influenced by many things, our DNA, lifestyle, activity, food we eat, stress, and of course the part on the lower left, our various exposures. And we can quantify all this now, some well, some clunky, but generally you can pull in the data. And of course we can quantify the impact of these by making deep molecular profiles. We've been hearing a lot about that today. And so, as many of you know, a number of years ago we set up something we call personal omics profiling. It actually started 12 years ago with me, and then for most folks, it's the smallest group, 109 people. We've been profiling for roughly 9 years, a little bit longer now for most. And we're sequencing your DNA and then making deep molecular profiles, mostly out of blood components. As you can see there, we do deep clinical testing, some advanced tests as well, especially in the area of glucose control, because I'm type 2 diabetic. And we do a lot with wearables, and that's where the Exposome steps in too. That started about 8 or 9 years ago. And one aspect is deep data collection, and the other aspect is longitudinal profiling. So we actually profile people every 3 months while they're healthy. And when an adverse event comes along, we'll take many, many more measurements. And we're doing this for lots of reasons. One is to understand healthy profiles, how does it differ between people, how does it change over time, so we can actually see how people are aging, which is kind of fun. And we're also trying to see whether these advanced technologies can be used to better manage people's health. And so just to summarize from the first about 3.5 years of work, we actually it's an older cohort, 53.4, although it's quite a spread, but we did discover roughly half the people, we learned something pretty important about their health. In some cases, it was a pretty big deal. For example, we caught some with early lymphoma, 2 cases of serious heart issues, and no one technology exposed us. Sometimes with genome sequencing, sometimes with deep data profiling, sometimes imaging, and so on and so forth. And it covered a wide span of areas. And I should emphasize that this is all pre-symptomatically. We found these things by profiling people while they were healthy. And then actually, there was follow-up to actually verify a lot of these tests. We have commercialized this, and actually the company that's done this, a more medical version, actually has had the same kinds of results. We caught people with early pancreatic cancer, for example, et cetera, et cetera. So the deep data profile has been pretty powerful. Now, of course, the biggest part that's missing out of this is how does it relate to environmental profiles and lifestyle, and that's where the Exposome kicks in. And so a number of years ago, we started bringing the idea of personal Exposome profiling, if you will. And we've really been concentrating on airborne exposures in 2 different areas. One is on the biologicals, shown on the left, and the other is on the chemical side, shown on the right. And so we built these exposometers, or I should say we adapted an existing one. Those measuring standard things like PM 2.5, temperature, things like that, where we actually, under the intake valve, engineered into a filter that captures particulates such as biologicals of pollen, bacteria, fungi. And underneath that, we put a chemical absorbent, tested a number of ones, and came up with one we like that measures basic chemicals that are in the air and absorbs those. And we analyze the biologicals, if you look at the lower left, by actually eluting off the filters, doing deep sequencing of RNA and DNA to see all the components that are there. And on the right, bottom right, we actually measure the chemicals by doing deep mass spec profiling, initially with an exactive, then more recently with these Orbitrap exactives, sort of things that Benedict talked about. I should say that's generation 1. We're using for the cohort here, generation 2. We've built a generation 3, although it's not out yet in scale, partly because of supply chain issues. So anyway, each of these is more powerful than the previous one, collects more per unit time, and gives us more information. And so from the Exposome, just summarize some early work, and then I'll spend more time on the recent work. But from the biologicals, we discovered over 2,500 species you can identify from the sequencing. This is just from the DNA. You can also get a large number from the RNA. And each sample would have about 300 species in it, and so I'll let you know what the raw species is at some point down the road. We'll see what that is. And then on the chemical side, we found what tentatively assigned 3,000 chemicals. It's a little tricky, as you heard from Benedict, at making these assignments, but then we go in for a certain set of them and validate. And so for 158, we have basically high confidence validated annotated chemicals, and they span a wide range of different things. You can see they're pesticides, pharmaceutical products, plastics are everywhere. I was surprised to see DEET, this thing you use to keep insects off, is actually found in all samples, although the amounts will vary from one location to the next. And the nice thing about this is we can actually quantify exactly what's going on in relative terms. So one of the first things we discovered is that this is just 3 people. I forgot to say, if we back up, our initial study was done on about 15 people, in a smallish study. This was 3 people who we profiled for a fairly long time. I'm P1, that's about 2.5 years of data. P2 is about a year data. P3 is roughly about 4 months of data. And basically what you're supposed to notice here is that every sample is a bit different. Okay, so depending what you sample, where, when, we can see what's going on, and you can see some exposures more bacterial, others more fungal, some plants, some animals, and so on and so forth. Many are mixed, of course. And then some of this is fairly reproducible in the sense that if you look at these, I don't really have a pointer, but those arrows at the bottom, every time I'm in Monterey, I get a fungal exposure. So you'll get very reproducible exposures if you go to the same location typically, not always. And we can quantify all this. At the end of the day, the bottom line is for what we can quantify, location is the #1 driver of your exposures, and seasons turn out to be #2. And I won't spend a lot of time on this, but you can show even people who live in fairly close areas, like in the Bay Area, each of those colors is a different person. If you look in the upper right, and people's samples will cluster according to where they live for the most part. And then at the bottom part, we can show there's a lot of seasonal exposures. This is just the biologicals, I should say, but you get the same result with chemicals. There are a lot of seasonal compounds and biologicals as you follow people over time. In fact, there's thousands of them. Okay. So, and we can then start correlating biologicals and chemicals, and they actually are some of them are highly coordinated. There's about 80 that I think we've identified now that you can identify. Here's an amusing one, which is pyridine, which is found in paints, or used to be, until very recently. It's found in many other compounds as well. It negatively correlates with fungi. So wherever you have a pyridine exposure, you won't have a fungal one, and vice versa. So you can make these kinds of fun correlations, and they actually have value. If you're allergic to mold, you might want to paint your house with a pyridine paint. You might die of cancer, but at least you won't be sneezing all the time. So those are the trade-offs you could do. That was a joke, didn't go over very well. Never mind. All right, so here's what we spent a lot of time lately with the pandemic, which is data analysis. So the ultimate goal is to be able to integrate external exposures with internal omics profiles and chemical, if you will, biochemical compositions. So again, we're trying to correlate what's going on in that upper left with all the metabolites, proteins, even your gut microbiome and so on and so forth, to see what kinds of correlations you can see. Okay, and these are associations, I want to point out. They're not cause and effect necessarily. There are ways of actually looking at that. We're not at that level yet. So what I'm going to tell you about are some of the associations. Okay, so what we've discovered, first we had to go in and annotate all the, if you will, toxins and xenobiotics and such that are actually found in our plasma. We spent a lot of time building a pipeline for that, much like Benedict has done. And so we can actually classify various compounds. I can tell you this is with a lower sensitivity instrument at the time. Only 11 compounds we found on the outside you'll find in people's blood, presumably because things turn over and such. Although we can find things in people's blood that we missed on the outside, and that's not surprising either, especially when it comes to skincare and pharmaceutical products. So in the end, we could identify something like 67, you'll see, annotated toxins or exogenous compounds, I should call them, that show up in your plasma. But even though that's a rather small number, you can see thousands of correlations between what's on the outside and what's on the inside. So even if they're not direct, there's clearly a lot of these statistical significant associations. And it's not trivial, it's a huge amount. Okay, and so this is a complicated thing, but I think this framework will be very, very powerful for how to do these kinds of analyses. So I would actually argue I'll take Jana one step further. I actually think we are here for doing multi-omics integration, and we could do this in a pretty sophisticated fashion. So just to explain this, I'm sorry we don't have a pointer, but if you look at that big purple bar, that is the biological exposures, and the orange bar at the top are the chemical ones. And these, every row, you can't see it, they're all blended because there's thousands there, correlates with something on the inside, meaning your microbiome from the bottom, that's proteome in red, metabolome in green, let's see what's next, microbiome in brown, and cytokine in that gray, and then clinical tests all the way at the top. So you can make these correlations, if you will, between the external chemicals and biologicals with all the different ohms, again, in a pretty sophisticated fashion. And this is all done in one person, me, actually. If you have enough samples, you can see how they're correlating, again, in a very robust fashion. And so it's a lot of fun, actually. You'll see, you can start seeing correlations between plastics and certain plastics and inflammation, carcinogens. That's probably one of the top markers, inflammatory markers on the inside that show up in your proteome, your cytokines, and your metabolites, and very specific ones. And they vary with the different, if you will, external exposures. I won't have time to get into this. But you can make all these different associations. And this just shows a few examples. It's hard to see where you see the red and green arrows. There's 6 major nodes, in this case, microbes in your gut microbiome that will correlate with biologicals and chemicals in our exposome. Again, it may not be a direct effect, probably isn't in many cases. Very few microbes on the outside show up on your inside. But the associations are very strong. There's some that are extremely strong that will show up. And again, many of these are related to inflammation. So the top 2 shown here, Rose borea and owl stripes, are actually highly connected to a lot of your external exposures. And this, again, is on me. This is the kind of thing we want to do on multiple people as we collect more data. And same thing, you can do this for metabolite. You can go through this for every single ohm and make these sort of associations. This is a metabolome. I think this one shows that, this one I flipped around, but you can show a lot of plastics and actually chemicals and other pesticides, it turns out, a lot of them have very strong inflammatory effects. And in this case, you can actually see where they map onto by where the metabolites normally function. And a lot of these are showing up in kidney and liver functions, a lot of the metabolites that are showing you strong associations with your external exposure. So this is the kind of thing we should be able to do when we get the whole bolus of data in for this cohort. And of course, this will be very, very powerful when you're analyzing pregnant women. Some of this data we think is clinically actionable in the sense that you can actually take clinical markers and make these same sorts of associations. So here's some associations with glucose and external exposures. Eosinophils, which are involved in allergy, that's an easy low-hanging fruit one. In my case, it actually shows up with eucalyptus pollen and urea nitrogen with kidney function and so on. So you can make, again, these sorts of associations. I want to take a moment to diverge, since I know we're doing stuff with wearables here. We're totally keen on this space. Many of you may know I have a whole giant team on this. We actually showed you could detect infectious disease from this, and actually we discovered my Lyme disease from a smartwatch and a pulse ox, actually, on an airline flight. And it turns out we think your resting heart rate is a great indicator of stressor. It's way, way better than temperature. And many of you know you don't get fever when you get COVID. I certainly didn't when I got it recently. And actually, temperature is a 300-year-old concept. I think it's a terrible marker for stress, such as COVID and disease. So anyway, we showed first that you could detect Lyme disease by accident, if you will, with the smartwatch, and later showed respiratory viral infections. We've gone on now to actually show you can pick up COVID 80% of the time before or at symptom formation. And the median for offline detection is about 4 days prior to COVID onset, but we have a real-time detection algorithm now. I'll show you that in a minute that actually does it in 3 days. This is one example. That purple day 48 is the diagnosis day. The symptom day was the red one there, day 47. But just with resting heart rate alone, you can actually pick up this illness 9.5 days in this case prior to the person getting it. These are very, very sensitive markers, and you only need 2 beats per minute to pick up this sort of thing. So again, I think this will be very powerful data as we integrate this into the HEAP consortia. And this is our real-time detection. I want all of you to sign up. I think I forgot to put the website here. Europeans are allowed to be part of the study. We have 10,000 people in the study. Just from the first thing we published in January, it works 80% of the time. It's not perfect, But it does work on asymptomatic cases. 14 of 18 symptomatic cases, we could pick up those illnesses, those stress events with SparBot. It's not specific for COVID, I want to emphasize it. A variety of stressors will trigger it, but infectious disease does definitely trigger it. All right, here's an asymptomatic case. I did put an example. I'm probably out of time. So to summarize, we've built methods and approaches for being able to measure external exposures, and with the wearables, things like COVID and other stressors. The exposome is very vast, but in spite of its size, we can make correlations. It's a big data project, correlations between the external and internal exposures. They are highly correlated on the outside, but they're also very correlated in what they'll do to your metabolic profiles and other profiles. And lastly, we have built, I wasn't going to get into this because I think you have a pretty adequate system yourself, but we have a nice study app for actually pulling in, running surveys and things right on an app that's open source. And then we have a whole cloud-based infrastructure that probably has a lot of analogies that Jim will, looking forward to talking with him further later. So, but you're welcome. All that's open source. I'm very fortunate. I have an amazing team who works on this. The 3 folks at the bottom, now a 4th, is all working on Exposome. Allison is the one driving all this for the HEAP consortia. I've done virtually nothing but give it the talk, and these guys do all the work. And then we have amazing people doing wearable and the general Exposome work in general. So happy to talk to all of you later. Hope I'm on time. Bye.
Oh, thank you very much. Really interesting.